So it's my great pleasure to introduce Polina Golland. She's a professor at MIT and also the director of the Computer Science and Artificial Intelligence Lab there, CSAIL in short. Right? Associate. Oh, this is not, but it's associate director. Okay, sorry, I misread, but maybe I can promote her here by saying this. She's like one of the leaders in medical image computing, and she's done great work with her students on image segmentation, population studies, shape analysis, and various other things. And I'm very happy to have her here today, and then also at Duke and NC State. And without further ado, let's welcome her, and she's going to talk about image imputation. Thank you.
Thank you, Mark. Thank you for a nice introduction. Good afternoon, and good afternoon to the two other vertices of the triangle, whoever is there in the rooms. So Mark gave me a very nice introduction. He promoted me to the director of CSAIL. I am associate director, ex-associate, as of last semester, and delighted to spend more time on the research. So what I want to tell you today, let's see. Yes, all those senior things. One thing I've never learned is how to give overview talks. So what you're going to get today is one slide of what we do, and then I'll focus on just one project that is the current favorite. And I was telling Mark that this is the first time I'm giving the talk on that technical topic. So we'll see. Okay, my group is working on understanding medical images and clinically relevant information that is captured by those images. I started by looking at shape, anatomical shape, and then very quickly segued into looking at regularities in pathology, which usually is something that we think of as something that doesn't follow the description of statistical models. But recently, we've been building statistical models of pathology. We've also looked at cases where anatomy defies shape modeling. For example, in congenital heart defects, the shape of the heart looks very different from the standard anatomical shape. As one of my clinical colleagues says, you don't get to be an adult with a heart like that, and so these kids need interventions soon after birth, and we are helping the clinicians to understand where the heart is and how to rewire it. And finally, recently, we got involved in a project in fetal and placental imaging where methodologically it's a little bit of everything, because everything moves in the scene, and you can't tell the mother and the baby to stay still like we do in adult imaging. So this is basically sort of a menu of projects that are going on in my group. But today I want to talk about image imputation. So before I tell you what image imputation is, let me tell you what the problem is and how we came to find we stumbled on the problem and then figured out the solution for it. So given that Mark and Steve and Martin are in this department, I suspect you've seen about 1 million images like this. This image is acquired as a cross-sectional slice through the head that goes like that. And if you take a stack of these slices, you end up with the whole volume. So in clinical studies, when we want to understand, for example, the effects of schizophrenia on the brain, the slices are placed so densely in a volume that the resolution with which we're acquiring across slices is exactly the same as the resolution within the slice. And in particular, if you take your volume and slice it like I'm doing in that plane, you'll get an image like that with lots of very fine detail about the cortex right here, the sheet of the fat under the skin above the scalp, and so on. But it turns out that almost no one acquires images like that in a hospital. So if somebody comes in with a prescribed MRI for clinical purposes only, then the radiologist or the clinician who prescribed the images skips quite a few slices in between. In particular, the problem that I'm going to talk about today is imaging acute stroke patients, meaning that we're doing the imaging as the person came into the hospital with suspected stroke, and the common practice is to skip 5 or 6 slices and acquire every 6th or 7th slice. And so if that happens, then the data really looks like this, where you're getting information across these thin planes, but nothing in between. So if I skip every 5 slices and acquire only the 6th slice, it's about 15% of all of the data. And radiologists, as well as practicing clinicians, so for example, stroke specialists, are very used to looking at the axial slices, as we call them, within the slice, and really quickly assess what's going on. But there is a problem with this, which is, well, and so then to visualize it, we'll use something like linear interpolation, and an image looks like this, which looks pretty terrible around here. All of the details are lost. And the doctors don't view this as a problem because they never look in this direction. They assess the state of the health of the patient by looking at the slices that got acquired. Is it mine? No, it's somebody calling. Well, I've never gotten a phone call during the talk. Hello? Sorry? I think it's a wrong number. Whoa, this is the first. Okay, and so the doctors look just in slices, and everything is perfectly fine. And so the situation we have now is that there are millions of images in the hospitals that are completely useless for research. And the reason why they're completely useless for research is because all the algorithms we've developed assume that there is isotropic sampling, or at least not too terrible of a sampling in the perpendicular direction. So, for example, most of the algorithms for taking these images and putting them in a common coordinate system or doing any kind of processing want to compute gradients of the image. Now, the gradients of this image are much more indicative of the direction of the scan than any anatomical features. And the reason we discovered this is because we started working with images like that and discovered that basically nothing works in these images that works beautifully in images acquired with isotropic resolution. And in particular, the problem that we were interested in is a problem of identifying genetic associations between, again, genetic code and white matter disease and stroke lesions in these patients who come in to the hospital with a suspected stroke. So the goal of the study was to take large collection of images. We, at this point, have something like 3,000 images. Put them into a common coordinate system and understand how presence or absence of white matter disease in certain location affects recovery from stroke, and what genetic code affects the development of white matter disease and potential risk for stroke. And we started by saying, well, what's the big deal? We'll just linearly interpolate the images and run the existing algorithms. About 20% of the algorithms fail what's called brain extraction phase, which is the phase, let me go back and show you, which is the step that basically takes the brain and extracts it from this rim of skull and tissue around it. And about 30% of the images failed the registration step, meaning the spatial normalization into a common coordinate system. And so here are the things we've tried. Interpolation doesn't work. We tried splines. It still gets the steps. Then non-local means is an algorithm that averages image patches that look similar, even if they could come from different locations in the brain. And again, I'll show you the results. It does as poorly as linear interpolation. And then another direction that we sort of thought about that is commonly used in the field for super resolution is to acquire a high resolution image set, subsample it, and create a dictionary effectively of low resolution image patches and high resolution image patches. And every time I encounter a low resolution image patch, I substitute the most likely high resolution patch into the image. The problem with that is that for many clinical cohorts, we have 0 high resolution images to use and represent pathology accurately in this case. So instead of using that, we came up with a method that after a year of working with this, I was telling Leonard, we recognized as imputation. So imputation is a procedure that was introduced by Rubin, I think in '76 in statistics, that attempts to estimate probability distribution for jointly distributed random variables, and then based on observed variable, tries to estimate the missing variable from the probability distribution. So that's exactly what we're going to do here. We will build a model for image structure, local image structure, from a large collection of sparsely acquired scans. So we won't require a single high-resolution scan. But what's important is that we have a large collection of brain scans, for example, in this case, if we're trying to reconstruct the brain, that were acquired all in slightly different orientation. And we'll see in a minute why that is important. And then we will use that model to fill in all of the voxels that didn't get imaged during the acquisition. So to understand, to see the difference between the interpolation results and imputation results, here's the example that I already showed you, and the zoomed in versions. Wow, your projector is really butchering these images. So here's a ground truth image of high resolution image of a particular subject. This is what happens if you interpolate a low resolution, not a low resolution, sparsely acquired image of exactly the same subject. You get these steps that completely mislead the algorithm, and a lot of detail in the cortex is lost. In contrast, if you perform image imputation using the model that was constructed using a large image collection, we recover the sort of nice rim around the brain that represents the fat, as well as quite a lot of detail that correspond to the true detail of the cortical folding. Okay, so now that I've given away the result, let's see how it's done. And the way I will do it, given the composition of my assumptions about the composition of the audience, I will first show you how it's done, and then for the particularly dedicated, there will be a slide that writes it out formally. So first, we will model the local image structure in a large collection of images. Let's see. So here are some images from that collection that I coarsely aligned to a common coordinate system. They have to be sort of roughly aligned so that somebody's nose doesn't show up where somebody else's ear is. But we've noticed that this procedure is actually fairly robust to misalignment in the data set. So I have a whole bunch of images, and if I put a small window around a particular location, then we see that a very different, somewhat different part of the anatomy shows up in the window. So here, the ventricle just barely scratches the window, whereas here, there is quite a bit of it. So we see that there is quite a bit of variation in any particular location in the image collection. So to account for that variation, we will put image patches sort of around that location. So we'll collect image patches from a neighborhood of the location. And I'll be much more precise in the second part of my explanation. For now, let's imagine that we collected a whole bunch of patches from a particular location in the brain. You can start seeing that some of them share the structure. In particular, this patch and this patch come from parts of the image where the bright is above the dark spot in the image. And for example, yes, right, these three patches come from that type of image, whereas these two patches might share a different structure in the image. So to understand that structure, we will cluster the patches based on their similarity. But of course, the most efficient ways to cluster, for those of you who have done it before, is not to look at pairwise similarity between the patches, but rather to create something that represents a cluster mean or cluster center, and then assign each patch to the cluster center that patch is most similar. And in fact, k-means does exactly that. It will take, k-means algorithm does exactly that. It will guess the cluster means, then assign every patch to the cluster center that is the most similar to, and then re-estimate cluster center and iterate. Right now, many of you are feeling smug, right? Because this is something that you've seen before. We're doing it on image patches here, but you've seen it done on vectors before. We actually splice the patches into vectors. Okay, but you've forgotten that all I get to see is this much. And so my patches look like this, not like what you saw in the previous iteration. However, if you wrapped your brain around clustering these patches, if I know where this mask is, meaning where the stripe is, which I do, because for any particular subject, I know exactly how I oriented them into the common coordinate system. With just a little bit of bookkeeping, I can still run my clustering procedure. In particular, suppose I guess these means, so the means are dense. So if I look at, for example, this particular patch, I can tell that it's much more likely to come from this cluster than from that cluster, because its intensity variation in the voxels where I acquired the image is much more consistent with this mean, with the red mean, than with the blue mean. And effectively, the algorithm runs exactly like that. It clusters the very sparse patches by doing a little bit of bookkeeping and comparing only the observed voxels with the estimated means, and then re-estimates the means. When you re-estimate the means, oh, by the way, so how come this guy shows up like this, whereas this guy is horizontal? It's because every patient was in a slightly different position in the scanner. So some people got acquired exactly like that, and some people got slices that are oblique compared to the sort of principal axis that we would like to put them perpendicular. And it's exactly the difference in these directions that will help us in the next step. Okay, so we've gotten the estimation. And I should say that the means are dense. We will estimate the dense means. And so again, you have to do a little bit of bookkeeping to know that when the patch here shows up as dark, whether it was the dark that was acquired, whether it was a dark voxel because it was never acquired, and never average it. Okay. So now we have a sense on how to build mixture model out of sparse patches in every location in the image. How do you impute based on this mixture model? Well, so if I have a sparse image that is acquired like that, and so here's a zoom out, a zoom in of that particular patch, I first find the cluster that most likely explains this image. So in this case, it's a purple cluster. Then if that patch belongs to the purple cluster, a mean is a good guess. But in fact, we will do something more sophisticated in a formal part of the talk. So I fill in all of the missing voxels and create imputed patch that is like that, and I plop it down into the reconstructed image in exactly the same location from which the sparse patch came. And then there are many different ways of collecting these patches into the right image. In particular, we reconstruct them in overlapping regions and then average the resulting reconstructions. And I'm happy to talk about this offline if anybody has questions. Okay, so let me summarize what we've done so far. Given a very large collection of sparse images that acquired at slightly different orientations, we will, in any particular location, we will cluster the patches so that there is a mixture model that explains the structure in the image at that particular location. And then given a sparse patch, we will find the cluster that explains it the best and impute based on that cluster. Okay, let's look at the formal model. So my generative model at the beginning is very similar to what you're used to when you do Gaussian mixture model. In particular, K will be my cluster membership that is drawn from a distribution pi that just tells me how likely each of the clusters is. Mu and sigma, so I didn't dare draw the sigma out for you here, right? So here's, for example, a mean, and here are the examples. And we say that the example patches, given the cluster, are Gaussian with the expectation mu and the covariance sigma. The reason why I don't dare draw sigma out is because we are working with three-dimensional patches of about 11 cubed, which is 1300. So I would need to draw a matrix of 1300 by 1300 to capture the covariance. We'll come back to it in a second. But actually, I don't get to observe these patches. Remember how what I got to observe is these sparse stripes through the patches where I know exactly what this matrix is that maps the dense patch into the sparse patch. In particular, all it does is it goes into the right indices and pulls out the values. so that this vector is much shorter than this vector, and it includes only the voxels that were observed. Okay, so let's look at this, and this is the part at which we were feeling smug before. So y and y observed are jointly Gaussian because it's just a linear transformation, which means that I can still run EM algorithm to fit this Gaussian mixture model. I just need to substitute this matrix to multiply both the mean and the covariance on both sides. And so it turns out, if you write it out, it's a nightmare. Because when you write out explicitly this Gaussian distribution, it has determinant of this matrix in the denominator. And that matrix is different for every single patch, because this matrix that determines the observations is different in every single patch. And basically, we haven't figured out how to solve the problem directly, because you have to integrate somehow the information over these inverse covariance, determinant covariance matrices. However, we will use the trick that oftentimes is used in data analysis. And in particular, instead of the matrix sigma that is unconstrained, oh, by the way, suppose you have all the patches. Sigma is still a nightmare to estimate because it's 1300 by 1300, and you're very unlikely to have enough samples to actually adequately estimate it. And so the usual trick used in modeling such structure is PCA, reduced rank covariance matrices. You've heard it by many names. And in particular, I will assume that there are loading factors X that are distributed from a normal distribution that multiply, some of you know this as eigenvectors of the covariance matrix, and add the mean. And this is what we call probabilistic PCA, where you add, there are many names to this particular model where we add a little bit of noise on top of your PCA analysis. So we know how to solve this problem if we forget about the clustering structure. And it's another EM algorithm. And it turns out that if you integrate that estimation into the EM algorithm for clustering, it just all nicely works out in a very efficient way. To run this optimization efficiently, you would have to basically use matrix cookbook and realize that you should never try to create pseudo inverses of matrix that is W transpose. Instead, like many of you who have done least squares estimate, it's W transpose W that you want to evaluate. It's the small compact matrix that you want to invert. Okay, and so we run the EM algorithm that takes all of the patches, keeps this observation matrix in mind for every patch. It could be in particular different for every patch, and reconstructs the parameters of the model, which I painted in this, I don't know what that color is. What do you think, Steve? Cyan. So pi, which is the relative proportions of each cluster, the mu, which is the mean of each cluster, W's, which are the eigenpatches, if you will, and this noise variance that is sort of a trivial parameter in the image formation. And we estimate those parameters, and the hidden variables is the class membership and this latent representation, the low-dimensional representation X that multiplies the eigenvectors to create our patch. And so, given the new sparse patch, we estimate both of the cluster memberships, meaning we estimate the most likely cluster to generate this particular sparse configuration and the latent representation, and then the dense patch that we estimate is a function of that latent representation and the parameters, the average, the mean, and the covariance, or eigenpatches, of that particular cluster. So as I promised you, it's not just the mean. We take into account the covariance structure of each of the clusters. Okay? So let's try to summarize. Given large collection of images that are sparsely sampled, we align them into a common coordinate frame using something like affine registration, which is much more robust than the nonlinear registration and oftentimes works. We then create subregions of the images that are overlapping. In our particular implementation, we use subregions of 21 cube voxels, and we overlap them by half of their size. For each subregion, we estimate one model, which is a mixture, Gaussian mixture model. And then given a sparse patch, we estimate which cluster generated that patch, as well as the latent representation and then imputed voxel values. I should say that the fully correct, let me go back and point out something. Given the observed patch, what you want to do is create MAP estimates, meaning to optimize jointly over, to maximize a mixture model, meaning a weighted combination of these representations over each cluster. And that becomes quite intractable. However, in practice, what we've seen, the posterior probability that a particular patch is generated by a particular cluster very quickly converges to either 0 or 1. And so even though in theory, it's a mixture distribution, it's a kind of mixture distribution where one coefficient is equal to 1 and the rest of the coefficients are equal to 0. And so that's why we can get away with just estimating that one particular cluster. And as I said, sorry, I'll go back. As I said, the way we reconstruct the entire image is we impute patches at sort of successive locations, and then we average the resulted imputed patches. Does this look similar to imputation in genetics, genomics? So I have to admit, we basically figured out what was wrong, implemented the algorithm, wrote out the model, implemented the algorithm, got the results, and only then realized that this was imputation. And so we were very excited to call it by its correct name. We called it reconstruction and restoration and many different things until somebody saw it and said, isn't it what they do in genetics, genomics? And that's how the name was born. Okay, so let's see how it works on two different image sets. The first image set is Alzheimer's disease neuroimaging initiatives image set. So this is an image set collected of healthy controls, patients who have Alzheimer's, as well as patients who are classified as mild cognitive impairment. It is believed to be a precursor for Alzheimer's. So quite often with aging, somebody who had mild cognitive impairment converts to Alzheimer's. So the patient, we have over 800 patients, over 800 brain scans in that initiative, collected by that initiative. So you could say, why are you telling us about it? These are the best images one could hope for to work with, because they acquired very carefully in a scientific study where the resolution is 1 millimeter cube, which is state of the art in brain imaging. And the answer to that is it gives us ground truth to validate our algorithm. So what we will do with these images is we will take all of the images available in ADNI, subsample them to 6 millimeter skip between the slices, which mimics our clinical image set that I will talk about in a couple of minutes. Then we build the population model. And so K is the number of clusters. And we found that just 5 clusters at every location was quite sufficient to capture the structure of the image locally. So this D is the dimensionality of every patch. This is when I mentioned that it's 11 cubed, which means 1,331 voxels vector that we're trying to model. And we found that the dimensionality of the latent representation was sufficient. So it was sufficient to set that to around 30. And we've done the usual scanning the values in the range and verifying that it's actually not that sensitive to setting of the number of clusters and the dimensionality of the latent representation. Then we once we've built the population model, we impute all of the images in that collection. And then we compare them to ground truth. So now we have imputed images as well as the voxels that were acquired through the scanner. We will compare it to several baseline models that are commonly used in our field. One of them is nearest neighbor. It's the most simple one. Quite often people then move on to non-local means, which I already mentioned before, as well as perform linear interpolation. And I've already said we've tried higher order splines, and it just basically doesn't improve the results. Okay, so let's look at the results. This is non-local means, linear interpolation, our method, and this is the ground truth. This is what the true images were before we subsampled them and subjected them to all of this torture. So you can see, and in this case, your projector is doing justice to these terrible methods. You're basically losing the contiguity of the rim, which is used by many algorithms to extract the brain and remove irrelevant parts of the anatomy. And much of the structure, so take a look at the temporal lobe. Much of the structure in these two methods are lost. So this is subject one. The imputation really does well on reconstructing the curved structure around the brain, as well as, so it produces somewhat blurry images compared to ground truth, but it preserves the areas of bright and dark and bright and dark. Basically, if you want to run a registration algorithm, when it looks at something like this, it thinks that the gradients are pointing along the direction of these steps, as opposed to perpendicular to the rim, as reconstructed by our method. And the second subject, this is just illustration of a couple of typical subjects in the study. And of course, we have statistics to back it up. This is statistics over all 800 some scans in the ADNI data set. And so this is nearest neighbor, non-local means, linear, and our method. And the mean squared error of reconstruction goes down as you go through the sequence of steps. This plot shows improvement over nearest neighbor for each one of these methods. So this is a pairwise comparison for each one of the subjects. The reason why we run it is because all of these methods have quite a broad range here, because for some images, the reconstruction works really well, even with linear interpolation. And for some images, it's quite terrible for linear interpolation. So here we're comparing for the same subject, nearest neighbor versus the other method, and we can see that our method again outperforms it. We also have comparisons of our method versus linear interpolation and each one of these baseline. Image imputation improves the quality of reconstruction as measured by mean squared error. We also looked at PSNR, which is another signal to noise ratio kind of measurement in these images for every single image in this collection. Sometimes the improvement is quite small because even linear interpolation gets it almost right. And sometimes the improvement is quite dramatic. Here are some examples of what happens when we run skull extraction. So in both of the methods, the skull extraction gets confused by those steps and decides that these parts of the skin and the fat under the skin are part of the brain, whereas again, image imputation extracted, and here's the ground truth. So image imputation tends to sort of create this almost convex hole, although I know that it's not convex, compared to the ground truth segmentation of the brain. We have also run experiments on basically brain extraction and spatial normalization, nonlinear spatial normalization of the imputed images, and it fixed it for every image that was failing it before once we run imputation. So let me show you examples of the stroke. So in the stroke image set, at this point, we're working with 700 images from Mass General Hospital provided by our collaborators. In this data set, we don't have ground truth. All we have is the input images, and we perform imputation on those. And that's why the fourth column of ground truth here is missing. And so you can, again, appreciate sort of the reconstruction of the area close to the skull by our method in both subjects versus what linear interpolation and non-local means do. They basically destroy the bandy kind of structure in those parts, as well as in other parts of the anatomy. So again, looking at the ventricles, so in contrast to ours. Our reconstructions are clearly blurrier than what you would get from high-resolution ground truth images. We basically don't have much intelligence to say about how to choose the parameters of the model to get sharper reconstructions. We can get sharper reconstructions, but then the noise increases. So basically, there is a parameter that allows us to span the range of how blurry versus sharp it is, and the trade-off is noise. And at least right now, I can't really tell you how to choose that parameter optimally. That parameter is the number of eigen patches that we're using to represent every cluster. All right, and so before I conclude, I would like to thank the graduate students and the postdocs working in my group, my fantastic collaborators at Mass General Hospital, and Mert Sabuncu recently moved to Cornell University. Adrian is interviewing for faculty positions as we speak. And of course, generous funding by several NIH institutes as well as companies. So to conclude, what I showed you today is an example of how we can take vast collections of images acquired in a hospital as part of the clinical practice and turn them into something that our algorithms for research images can run on. This promises to dramatically change how clinical research is done. Instead of recruiting 50 patients in a particular study and expending lots of effort to acquire perfect images of those 50 patients and risking not representing anatomical variability of the entire population at all, our approach is to take large collections of images acquired at the hospital that actually capture all of the anatomical variability present in the patients and perform image imputation and then run all of the algorithms that our field has been building for those beautiful images acquired by studies like ADNI. We hope that this approach will yield new clinical insights about diseases, how they affect the brain, how genetics relates to the symptoms or to the phenotypes that we observe in the images. Thank you.
Any questions for Polina? Okay, remote people first. Any questions from Duke or NC State? Duke, NC State, are you there?
Maybe they called before.
Maybe they called, I don't know.
So this is a question from somebody who does not work in medical imaging. So what if the problem is between the slices? You know, your resolution is 1 and 6. I mean, if the problem is smaller resolution than that, what is the hope?
It's an excellent question. Thank you for asking it. So the question is, what if the relevant part of the anatomy is between the slices and all hope is lost then? Because you can reconstruct all you want, but effectively what you're reconstructing is some average behavior of a population. And so the answer to that is we don't propose for example, use these images for detection of the disease. Quite often what happens is that what you're trying to do is estimate total volume of the disease or spatial distribution of the pathology. And in those cases, imputation provides effectively a scaffold. It enables us to take all the images, spatially normalize them very well, remove all of the imaging artifacts, and then measure only from the voxels that were truly acquired. But this approach would have these measurements in the common coordinate system of all of the patients combined into one atlas. Yes. Yes.
Yes, can everybody hear me now? Yeah, it might be a naive question, but what happens if you apply the method again to the image that was uplifted?
So the resulting images are blurrier than the ground truth. I think some of the information is truly lost because effectively we're representing the population as a mixture model as opposed to something very, very sophisticated. So I don't expect that if you run it again, it would get sharper. Because, you know, we can't produce sort of sharp details out of thin air. And in our experience, basically increasing the number of eigenpatches, those vectors W that multiply the latent representation, increases the sharpness. But then you start getting sort of that very sharp noise, salt and pepper kind of noise in the images. And right now, I don't know how to fix that. I think there is, this is starting to look like me. Excellent.
So I'm just wondering, why did you decide to choose K, a number of clusters, to be the same at every location in the brain?
Again, another excellent question, because we were drowning in decisions. Okay. Right? Basically, you have to decide, so you have to accumulate patches from a region to actually get a rich representation and to compensate for misalignments in the data set. So how big a region, how big a patch, how much to overlap the region, how many clusters to use. And at some point, it became sort of unwieldy, and so we said enough is enough. But I completely agree with you that it would make sense to have one higher dimensional cluster and another lower dimensional cluster, and we just don't know how to handle it, how to make these decisions intelligently.
So also I want to, like, what is your thought about like maybe using your method for having like quick annotations of the brain, so you can apply the same principles on having someone to manually segment like few slices, yes, and then do the interpolation the way you do to get like a quick segmentation of the brain that can be used in deep learning, for example.
So what I would do is not that. What we are doing now, so, sorry, I forgot to mention this. So this is running now at MGH, at Mass General Hospital, where they're leading this 3,000 patient study. So the image imputation is being used to bring all of these patients into the same coordinate frame, to skull strip, to intensity normalize, and then make the measurements in that coordinate system. So what I would do, sort of along the lines of what you're proposing, is I would build very accurate alignment after I've imputed of that patient to an atlas. I can have the atlas exquisitely annotated and then just map the segmentations onto every patient.
Okay, thank you.
I think it just promises better accuracy than having people do quick segmentations. So we do get questions, why don't you use deep learning to learn how to reconstruct high-resolution image, sort of fill in all of the voxels? I want to remind you, there isn't a single high-resolution scan in this exercise. So in order to do anything with supervised learning, you have to give it examples of what the answers look like. We learn those examples in a completely unsupervised way from only sparsely acquired images. Steve. And as the mic traveling, I should also add that if you look, let me go back to the model. If you look at the model, so I can show you the mean, and these vectors represent eigenpatches. Another way to think about it is that I have a structure for the sigma. In this slide, it's hidden, but I'm estimating sigma. And by looking at the slide, I think it should be obvious that you want to acquire every patient in a dramatically different direction so that you get samples of 2 pixels co-occurring in these sparse images. Our directions are sort of a little bit floppy, but they don't go dramatically perpendicular to each other. However, if you start acquiring images that dramatically disagree across patients, then the doctors don't know what to do with these images. So it's a bit of a dilemma, that what needs to be done for a computer algorithm is actually different from what people would prefer to interpret these images. Steve?
I'm going to ask a relatively unsophisticated question and then a more sophisticated one.
Okay.
Okay, so 51 years ago, when I was doing my dissertation at Mass General Hospital, I was concerned with taking blurry, noisy images and restoring them. That methodology has developed significantly over the years, a whole lot from what I figured out back then. But ultimately, in today's imaging environment, there are ways to sharpen noisy images and get some reasonable results. What prevents you from incorporating that kind of thinking on your blurry, noisy images?
It doesn't work. It's just that simple. We spent the first 6 months trying these tricks and failing over and over again. We also talked to everybody who published recent methods on sharpening the images. And effectively, when they say 6 millimeter,
it doesn't, I'm not, I thought you, sorry, but either I didn't ask the question clearly, or I think maybe you didn't understand. I'm talking about after the imputation.
I can sharpen them, but at that point, it's sort of uninteresting.
Okay.
Yes, yes, you could sharpen them, but at this point, these images go through spatial alignment, at which point I throw away all the imputed voxels and only measure the acquired ones. Yes, but you could.
Okay, the somewhat perhaps more sophisticated question. Perhaps you're aware of the low rank subdivision, low rank analysis method that Mark is working on to help find pathologies, essentially. And I was wondering about the application, and just musing about the application of that on the imputed images. Maybe that's too much to scratch your head about to answer right now.
But I'm happy to do it over time and get back to you. Yes. So, I mean, like, this is a low-rank model, right? So basically what we're saying is that the covariance matrix is approximated by a low-rank matrix where the rank is the number of these components, the small d, plus a little bit of regularization in the form of the identity matrix. I don't know anything about what Mark is doing with low rank.
We'll talk about that at the
end. And so we can discuss this over dinner. But I would love to know.
So for each one of these patches,
yes,
there's actually linear cross sections for which you have truth. So what is, have you quantified the area at the subset of the patch for which you know the truth? And is there a parametric model that you could modify your patch, like residual eigenvectors, to try to minimize the error in that fit?
Oh, you're giving me ideas. So here's what we have thought about and done. In this model, I estimate the cluster that produced the patch, the latent representation, meaning the loading factors on each of the eigenpatches, and then I reconstruct it like this. So some of these y hats were acquired. And what we noticed is that, of course, it gets smoothed over those as well. And so what we have tried is estimating it only in the missing voxels. And then it doesn't look as nice. But this is interesting. So basically what you're saying is that build it into the estimation of the population model, that the data has to agree on the acquired, sorry, the prediction has to agree on the acquired pixels. That's right. Yeah. I like this. We haven't thought about it. We sort of struggled with what to do with the pixels we've already acquired and tried it both ways. And the images reconstructed for the whole patch look better. But I like this idea. We will try. Go ahead.
Thank you. I think your work is interesting. And,
thank you.
Yeah. I think to achieve the membership between patches with your model is important step. And the model, to achieve a proper model is, I think, is the key step. But the patch size and the overlap size can affect the performance of the model. My question is, how to say we achieve a proper model? How to evaluate your models?
I got it. So what you're asking, let me just make sure I understand, is I've given you all these parameters, including the region size. How do you know that this is any better than other setting or so on? So we beat this model to death. There is a paper that is going through revisions in Transactions on Medical Imaging that sort of addresses it. So here's our answer to it. We've done fairly exhaustive search over the space of parameters, and it's a minimum, but a very shallow one. So if you set the number of clusters to 8, the results don't change qualitatively. If you make the regions 25 instead of 21 or 19, it doesn't change the answer that much, right? So I actually feel more confident when I have setting of the parameters that my answer is not sensitive to than proving to myself that I found the best setting. Because if that best setting is very fragile relative to the changes, I feel much less confident.
Thank you.
So for this MGH study that they're using this for, so what are they in fact measuring there? And is what they get out of this imputation, is this, they're happy with the results, or are there any issues with it, or?
I need to show you a slide.
Okay.
And I want to cover up and look for the slide. Does it work? Perfect. Here we go. So what they're interested in, so these are many examples of these images. And in yellow is manual segmentations, and this is our automatic segmentation of what's called periventricular white matter disease. There is lots of hypotheses about what it is. The most kind of like the most robust belief is that it's microbleeds created when capillaries burst effectively. And we all accumulated with age. So this is a particularly bad case, you see. So they're called white matter hyperintensities in these images because they just show up brighter. And this is all segmentation of stroke lesions in the same images. Here's an automatic segmentation of a stroke lesion that shows up in the same image. So the goal here is to measure how much white matter disease there is, as well as where it is, and to understand the relationship between that and genetics. And the reason why it's important is because there is lots of suggestive evidence that this is a cerebrovascular health index, basically, in some sense. In that the people who have a lot of white matter disease, when they get stroke, they recover much worse than people who have very little of it. And so there is lots of interest in the stroke community to understand white matter disease, its distribution, its relationship to stroke lesion, stroke recovery, as well as the genetic factors that underlie it. And so what we do is, once we bring these images into a common coordinate frame, we do model-based segmentations of the white matter disease and only measure where required.
Any other questions?
I have one question. I was wondering, is your method able to deal with images with pathologies? Because one patient have a specific location with the pathology, the other patient won't have.
All of these. Each one of these has a pathology in it, right?
So
basically every patient, every subject in our study is a patient.
Yes. So when you impute each patches, for example, one patch for this patient probably contain the pathology, while the other patient don't have. So you still.
That's right. And so what's going to happen then is that if, for example, this particular patient, we're trying to impute slice that is very similar to here, it won't have the brightness like this one does. So it will pick the cluster that doesn't have the brightness in it.
Mm-hmm.
Does that make sense? But overall, I'm not trying to imagine white matter disease where there isn't one or where there is one, right? Once I've brought these images into a common coordinate frame, I will not touch the imputed voxels because I myself don't believe that we are measuring, we're imputing pathology correctly.
Okay, thank you.
It would seem to me that MS, multiple sclerosis lesion analysis, would submit very nicely also to this technique. Moreover, you have somebody at the Brigham, Charles Gutmann,
right,
who has lots and lots of images for that. Am I right that that's a possible area of application?
So the problem with MS patients is that they're happy to go into the scanner and sit there until the image is perfect. So we went through this phase of about a year trying to solve this problem, and really like nothing worked out of the box. We had to figure out segmentation methods, this imputation helped, and so on. And we would go to the conferences, present it, and say, we're segmenting white matter lesions. And all of the MS guys would say, what's the big deal? And it's because they have 1 millimeter and sometimes even 0.8 millimeter isotropic images. So at which point free surfer works on these images?
Is there anybody at Duke or NC State who has a question? I suppose not. Well, then let's thank Polina again for a very nice talk.
But I think it doesn't matter. All right. Thank you.
Hey, is anybody here faculty by any chance?
submitted by